Welcome back, guys. Thank you for joining us. Before I start, I would like to send a vote of thanks to the following people for the great job they have done in training teachers and also giving resources to teachers so that they can send these resources to the students so that they can pass their exams. All of them are facilitators and they really have done a great job and they have provided us with enough support. We thank you so much. May this message reach you where you are. Madam Susan Fais, I don't know whether I've pronounced it correctly, is an Umarus moderator. Madam Redin Kuruga, this one is a chief marker for paper 2. Vadika Maharaj is a facilitator, that part of Westerland. Van Wyk Griselda is also a facilitator to the side of Sedibeng. Madam Nontlantla, this is also another facilitator for the side of, and then we also have my facilitator also, who is Fauzi Ramadan from Johannesburg West. Please don't forget that you can download these notes. You can download a memorandum and the notes using the link below. Don't forget to download it if you need them on your phone so that you can also practice. If you have any challenge, communicate to us. We'll try to solve those problems so that we see that every student in this country can pass. All right, today we are going to look at what we call human reproduction. M.Saidi, as usual, taking you over this subtopic. All right, if you get this diagram, what is the first thing you need to know? What is the first thing you need to know if you see such a diagram? Diagram below represent endocrine gland. Endocrine gland and events that take place in the ovary during menstrual cycle in human. In other words, gland A produces a hormone which is used in the ovarian cycle. We have only 2 hormones based on our syllabus which are being released from the brain and then they work in the ovarian cycle. The first one is luteinizing hormone. The second one is follicle stimulating hormone. So it means that those hormones which are going down must either be luteinizing hormone or follicle stimulating hormone. Don't forget to write the correct spelling. So you're saying gland A, structure B, follicle stimulating hormone. Basically, now we see that this is follicle stimulating hormone. If this is follicle stimulating hormone, automatically this one is going to be luteinizing hormone because we said you only have 2 hormones and follicle stimulating hormones, they have talked about it. So the next hormone must be luteinizing hormone. And then what is this? This is the mature Graphene follicle. This is the ovum and the process which is taking place here. It is ovulation. And then once ovulation takes place, you go to what we call corpus luteum. So this is the empty Graphene follicle which has turned into corpus luteum. So basically we have seen it. We said this is a mature Graphene follicle and then this is the developing follicle. These are the developing follicles. All right, let's start to answer the questions and then we see. Saying gland A, we have seen it that gland A is pituitary gland, or sometimes we can call it pituitary gland or hypophysis. Pituitary gland or hypophysis. Then structure B is here, which is the Graphene follicle. So we prefer to say mature mature, because even this one is Graphene Follicle, even this one is mature, is Graphene Follicle, even this one is Graphene Follicle, that one is Graphene Follicle. So what is the difference between this and this? So the difference is this one is mature, ready to produce the ovum. So it is mature Graphene Follicle. It's mature Graphene Follicle. And then structures, process C, we have seen it. Process C, we say that this is ovulation. And then we are saying that structure D, we say this is corpus luteum. Corpus. You don't forget between corpus luteum and corpus callosum. Corpus callosum is found in the brain, while corpus luteum is found in the reproductive system. So we go to the next question. They're saying that structure D, you have seen it, state the effect of estrogen levels in the blood if gland A stops secreting follicle stimulating hormone. So if they're saying the effect of estrogen in the blood if gland A stops secreting follicle stimulating hormone. So basically you have to know what is the effect. It's 1 mark, but you have to know the criteria behind. What is the effect of follicle stimulating? What's the function of follicle stimulating hormone? So the function of follicle stimulating hormone, the answer is already there, is hormone that stimulates the Graphene follicle. That's the answer already. So for the function of follicle stimulating hormone. So if the Graphene follicle does not develop, and we know that this Graphene follicle is the one which produces estrogen. So if Graphene follicle has not developed, then the level of estrogen will not be produced. Therefore, the level will not increase. It will either remain low or it will decrease, because the one which was there has been used up. So it will remain low or it will decrease. Then what state 1 function of LH? LH, we said it's the luteinizing hormone. It has 2 functions. The first one, it triggers, it causes this process to occur. It triggers ovulation or stimulates ovulation. Number 2, changes the empty Graphene follicle, this empty Graphene follicle, into corpus luteum. So they only want 1 function. Then when we go to another structure, it is for a male. The other one was for a female. Now we are going to the structure for the male. You need to know how at least to label. This is vas deferens, yes. And then this is epididymis. This is the outer part of the testis, which is the scrotum. And then this is the testis. So now let's label. Let's label based on the question. Part A, we have seen it is testis. Part B, we have seen it is epididymis. And then part C, we have seen it that it is scrotum. Don't forget to write the correct spelling for epididymis. Yes, epididymis. And also the correct spelling for testis. So test is 1, né? But testes if there are many. Testis if it's what? If it's 1. And then this one is scrotum. So describe the process of spermatogenesis. I explained this the other time when I was describing the paper of 2022 for an exam for June. And then remember that we are in a period of, you have to follow what you call the examination guideline of 2021. So if you follow the one for 2017, then it won't give you some of the things will be, find them changed and then not that much detailed. So here what you need, however much the book is showing you a lot of things, Please follow this. If you also use our book for the distinction material, still you can find it correct. So what do you write here? The process of spermatogenesis under the influence of testosterone. So it means that that's the hormone. That's the what? The hormone responsible for development of the sperms. Under the influence of testosterone, the diploid cell, so the diploid cell, the cell which has 2N, will undergo meiosis. So also you mentioning the word meiosis, we shall give you a tick. Found in the seminiferous tube. We need to know where is it found. Found in the seminiferous tube to form a haploid sperm cell, or to form haploid. So basically you have one cell which is 2N will undergo meiosis. Here I remember that you form two kinds of meiosis. Meiosis 1 and meiosis 2 will undergo this. Under the influence, this cell will undergo meiosis. But what causes it to undergo meiosis? It is the testosterone. That's why, where is this testosterone and this cell found? They're found in the seminiferous tubes. How do we call these cells which select themselves out to undergo meiosis? We call them germ cells. What do they form? They form the sperms. So basically, that's it. The germ cell will undergo meiosis under the influence of testosterone in the seminiferous tube to form a haploid sperm cell. Or you can say under the influence of testosterone, the diploid cell, that is the germinal epithelium, will undergo meiosis to form the sperm cells in the seminiferous tubes. So we need to know the hormone. We need to know what will undergo meiosis, that is the diploid cells. The beginning is 2N, and then where is it found? It's found in the seminiferous tube, and which process will undergo, which is the process is meiosis, and what will they form, which is what? Which is the sperms. That's what we want in this regard. If they ask you to explain spermatogenesis, the same story when you go to oogenesis. Then we go to another question. Explain why a doctor would advise the man to wear underwear that is not tight. So remember that whenever you are tight, the pressure increases. And the moment the pressure increases, in most cases, the pressure is associated with the high temperatures. And then if the high temperatures are there, then definitely the temperatures are going to exceed the normal temperatures at which sperms are being produced from. Therefore, it means that it's going to lower the sperm count. That is the basics behind that. But now, how do you answer that in the exam? Maybe you can say that the tight underwear will put testes close to the body. When you put it in tight underwear, it will pull the testes close to the body. Yes. And then this will cause testes to have high temperatures or too much temperatures. Yes. And then this will affect the sperm maturation negatively. Don't say affect the sperm maturation. How? Negatively. It means that it will lower. Yes, it will lower how many and the rate of these sperms maturing. So if there are less sperms which are being produced, it means that the number of sperms at the end will be low. That's why we are saying that it's going to reduce the sperm count. Or you can explain it in any other way, as long as you talk about that the testes are going to be a little bit close to the body. And then once they are close to the body, they're going to have higher temperature, more than the temperature needed for the sperm production, and then this will influence the sperm maturation and production, and hence the low sperm count will come in. So please don't forget to explain that. Then they're saying that during vasectomy is to cut these testes. When you cut this vas deferens is what you call vasectomy. So they're saying that during vasectomy, explain one reason why a man who, number one, does not want to have children will choose to have vasectomy. This vasectomy does not mean that if your vas deferens are cut, you cannot undergo sexual intercourse. You cannot have sex. It does not mean that, because the penis to erect is not connected to the testes. The penis to erect is connected to the blood being supplied to the penis. This person, the blood will be still being supplied the tissues of the penis. Therefore, this person can erect and can have sex. If you cut the vas deferens, the only thing vas deferens is going to do is that the semens which are being produced, remember we have the gland of prostate gland, the Cowper's gland, and the seminal-vesicle, they will produce a fluid, and that fluid will not have sperms. So there will be no sperms, but that fluid will be there. The fluids will be there, but it will not have what? Sperms. And if the sperms are not produced, so what will happen? It means that the semen which are produced cannot fertilize the ovum. Hence, there is no child. But the person can still have the what? The sexual intercourse. So what is the answer in the exam? You can say that there will be no sperm in the semens, therefore no fertilization will take place. So no sperm, no fertilization. No sperms, no fertilization. So we will have no fertilization. Yes. So if you don't have sperm, no fertilization. Part B, had a vasectomy, is still capable of ejaculation. Why is it that this person is able to produce other fluid from the body when you are ejaculating? Is because I've told you that you still have the other glands. Look. So vasectomy is here, and then the glands are here, and then the penis is here. So you have the vas deferens, you have the Cowper's gland, and then you have the prostate gland. So other fluids will come from those glands, and they will make the ejaculation possible. Hence, you can still enjoy sex. All right. Question 12. Another question is about still reproduction. The first one was about female. The next one is about male. Now this one is both male and female. All right. They're saying diagram below represents structures of an ovum and the sperm. So you have to know how to label. At least to try. What is this? This is the jelly-like layer. What is this? This is the cytoplasm. What is this? This is the acrosome. What is this? This is the nucleus. What's this? It's a middle piece or mitochondria. And then what is this? This can be a tail or flagellum. So identify part A. They have said it. Gel-like layer. Part B, cytoplasm. Part C, acrosome. You have to know the function of each. Name the process involving meiosis that lead to formation of the ovum. You see, now they want formation of or production of the ovum. So because it is ovum, so it's gonna be oogenesis. And then I'm saying write down only the letter of the part of the sperm that enters the ovum. the letter of the part which enters the ovum. So basically, if you look at this sperm during fertilization, this part will remain out, and then this part will be used to digest the membrane of the ovum. It is only this nucleus which will enter the ovum, and then the haploid nucleus of the sperm will fuse with the haploid nucleus of the ovum, and then a diploid zygote or diploid zygote is formed. So basically it is going to be the nucleus, and which part is that? Is part D. Part D, yeah, is the one which is going to be used. Then they're saying that write down only letters of the parts that enable the sperm to move towards the ovum. So if we look at what provides energy, we shall see that the middle piece and the flagellum or the tail are the one which are used. So the middle piece will provide mitochondria, and mitochondria is responsible for energy. And then this one, it will provide propulsion force, the forward force, so that now the sperm can move forward. Yes. Then they are saying that now we are going to process of fertilization. The schematic diagram below shows a human ovum that is about to be fertilized. The diagram is not drawn to scale. All right, they're saying, what is part A? We saw that part A is the jelly-like layer. What is part B? Remember, this is part B comes up to here. Yeah, up to there. That darker layer. Remember, this is a cell. And all cells, they have a membrane which protects them, allow gases, substances to go through, which we call cell, the membrane of the cell. The membrane of the cell, which we call cell membrane. So the nucleus is there. Remember, this is an animal cell. Therefore, B becomes the cell membrane. What is C? C, you have the fluid there, which we call cytoplasm, the fluid of the cell. And then what is D? D, you see that is the nucleus of the sperm. Nucleus. And then what is E? E is the acrosome. And then what is F? F is the nucleus of the ovum. And then what is G? G is the middle piece. So basically we have labeled it. So A, they want A, we have seen it. A is the gel-like layer. B is the cell membrane, the membrane of the cell, or plasma membrane. Then C is the cytoplasm. And then F, where is F? F is the nucleus of the ovum. So now give the letter and the name of the part that contain mitochondrion. We have seen it that mitochondrion are being stored in here. If one is mitochondrion, if there are many, so the answer is G, and the part is middle piece. Contain enzyme required for penetration of the sperm into the ovum. So it is this. It has the enzyme. We don't need to know the name of the enzyme. Then the part, we are saying that it is E, and the part is acrosome. You don't need to know the enzymes because this is not our syllabus. And then they're saying C will enter the ovum during fertilization, and that is the nucleus of the sperm, which is part D, nucleus of the sperm. And then here we have the structure of the sperm. You're saying that A, B, See, now we have tried to answer questions concerning about the sperm. So there is no way they can bring a question you have never seen about the sperm, about these structures of the sperm and the ovum. So we're gonna go to even the graph. We also explain. We're gonna explain all topics in such a way that there is no way you can fail this. There is no way. And if you do this, you go back and listen to this before the exam, before you write the exam, look, and at least you are reading like this, then automatically just guarantee yourself a distinction. All right. Then they're saying that what is part A? We have seen acrosome, part B, nucleus, part C, middle piece, part D, the flagellum or the tail. They are saying that identify B. We have seen it. We have seen it. It is nucleus, part D. We have seen it is the tail. Then they are saying that explain one way in which the sperm cell is adapted to ensure effective movement. Whenever they talk about movement, you talk about the parts which help the sperm to move. And we only have two parts which help the sperm. Adaptation is the part and the function. The first one is the tail. Yes, the tail. This provide the forward force or provide the propulsion force. So it allows movement to take place. So basically this is for force. And then number two is the middle, this part, middle piece, middle piece. This one provides it has mitochondria which are responsible for production of energy. Yes, production of what energy? Explain the consequence for reproduction if the sperm did not have part A. What is part A? It is the acrosome. So what is the function? You first ask yourself, what is the function of the acrosome? The function of acrosome is to release enzymes which are used to digest the membrane of the ovum so that the sperm can penetrate during fertilization. So the major function is to contain enzymes which digest the membrane of the ovum so that the fertilization can take place. So if part A is not there, it means that will not take place. It will not happen. So basically, no acrosome will be present. So the word no acrosome, yes. And then therefore there will be no enzymes present and the sperm will not be able to penetrate the ovum and therefore no fertilization. If there is no acrosome, there is no enzymes and the sperm will not be able to penetrate the ovum and no fertilization will take place. At least you will have something like 4 marks out of 3 and then we'll be able to obtain those marks. Guys, please watch these videos. Forward them to others so that they can also benefit. If you have a question, we are here to try to answer those questions, because some of these questions we do as a result of other students trying to ask for help. Now you have another question. Process. This is process from the sperm to the development. Now I think now we know the structure of the sperm. You see that they keep on repeating these questions over and over and over again. So what makes you not to memorize it? What makes you to go to the exam when you don't know this? I don't see any reason for that. Acrosome, we have seen it. Nucleus, we have seen it. Mitochondria or middle piece, we have seen it based on the question which is being asked, most especially if the line is not clear. Identify A. We have seen it. Acrosome. Name the organelle found in large numbers. In part C, we have seen them. You see, it is mitochondria. Then they are saying give the numbers 1, 2, 3, 4 only on the diagram representing morula. What is a morula and what is a blastula or a blastocyte? Blastocyte or blastocyst, the way you pronounce it, as long as remember. A morula is just a ball of cells. A ball of cells. this ball of cells has no hole at all. So you look for something which is like that. And then a blastula is a hollow ball of cells. It must have a hole. Is a hollow ball of cells. So you look for something which has the hole. So basically, morula is gonna be number 3. Is gonna be number 3, and then blastula is gonna be number 1. And then they're saying the structure that will implant in the nucleus. Which structure will implant? The moment you form a blastula, is gonna form finger-like projections, which you call chorionic villi, which are used for implantation. So basically, it's gonna be number 1 again. Yes, so it's gonna be number 1. Give the letter and the name of the part that will enter the ovum during fertilization. This is the second time they're asking the same question. So it is B, and it is what? Is nucleus. Then they're saying that name the type of cell division that will occur to produce the structure in 3. So the moment you have a sperm, yes, and then you have the ovum, nucleus. This nucleus comes here, and then you have a nucleus of a sperm and a nucleus of the ovum. You form a zygote. This zygote will start to undergo mitosis repeatedly to form a ball of cells. The ball of cells, which we call morula. The morula will still continue undergoing mitosis and starts to form the hole in the middle or in the center, and then forms a hollow ball of cells, which we call the blastula. The blastula will form the outer layer, which is going to call chorion, and the outer layer is going to form finger-like projection, which we call the chorionic villi, for implantation to take place. So, which process results in the multiplication of these cells? The process is called mitosis. So I've explained the development of the zygote until implantation. Let me repeat. Yes, we have seen that sperm fuses with the ovum to form a ball of, to form a zygote. You see, these are 2 nuclei, and then form a zygote. A zygote, you undergo mitosis to form a ball of cells. A ball of cells will continue. This ball of cells, we call it morula, and then this ball of cells will continue undergoing mitosis to form a hollow ball of cells, which we call blastula, which forms the outer layer. We call it chorion, and then the chorion forms finger-like projection, which we call chorionic villi. Basically, this is 1, this is 2, this is 3, it's 1, it's 2, it's 3, and then it's 4. Then the process I was trying to explain is here. The diagram probably represents a sequence of events that may take place inside the human female reproductive system. Identify the process taking place at 2. Where's 2? You see? Zygote from ball of cells, which you call morula. Diagram 1. Diagram 1, you say sperm fusing with the ovum, you wanna form the zygote. So the process is fertilization. State the type of cell division that takes place at 2. Here it is mitosis. And then they are saying that name 2 functional extraembryonic membrane that are produced by structure C. They are saying functional. It means that something which is functioning. We have 4 extraembryonic membranes. We have the chorion, allantois, yolk sac, and then amnion. But in this case, in humans, we only have the chorion, and the amnion. amnion. Yes, the chorion and amnion. We don't have the yolk sac. We don't have, because our egg doesn't have the yolk. Yes, and then we don't have the allantois, because this excretion does not take place. It does not deposit the wastes in there. Excretion will take place in the placenta or through the placenta. That's where the gases are being removed, and then useful substances will come in. So it takes place. So the placenta will take the role of the allantois in case of the amniotic egg. Yes, identify the stage of development indicated by, okay, A, what is happening there? That is a zygote. And then B, it is a morula. And then C is a hollow ball of cells, which we call blastula. Yes, or blastocyst. Make sure that you write the correct spelling. Name the part of the female reproductive system where the events in the diagram above usually take place. Where do we find this? They occur in the fallopian tube, because fertilization will take place in the fallopian tube. Will take place in the fallopian tube. If this is a simple structure, yes. Fertilization will take place here, and then these events will keep on moving until they reach here. Once they reach here, then implantation will take place on the walls of the endometrium. But all these events we are seeing, they're taking place from the ovary. They are moving. They take place along the fallopian tube. Give the chromosome number of cell at A. Chromosome number cell at A. Remember, sperm is haploid, ovum is haploid, and then when they form 2N, which is a diploid. The remaining ones are diploids until they produce a sperm again. So they're saying, give the chromosome number of the cell. They want the chromosome number, and they say that this is a human diagram. Therefore, if it is 23 here, 23 here, therefore this is gonna be 46 chromosomes. But this is for an individual who is normal. But here they're saying that if a child had Down syndrome, so remember, Down syndrome, you have an extra chromosome on chromosome number 23. So it means that it's going to be 46 plus an extra chromosome, which makes them to be 47. So the answer is going to be 47 because of the condition of Down syndrome of an extra chromosome on chromosome number 21. So we're just going to break, and then just few seconds we're gonna come back, and then we explain the remaining subtopics. We still have a lot of questions which you have to exhaust, so that by the time you write the exams, you can do this. You can do this any day. Tomorrow, make sure that by tomorrow everything is ready. At least you go to the exam and then you write. I thank everybody who is watching. The next episode, we will talk about those people who are always watching and commenting and asking questions. We thank you so much. At least you made us believe that what we are doing, at least, is helping some people outside there, and this gives us more energy to do more and hunt for more for you. Thank you very much for watching.